uh, we already discussed all the steps of the mitosis division, and also the introduction of the DNA, nucleic acid, we already discussed. When we see the DNA structure, so DNA is composed of the two helix or two strand, and these helix, I already told you, the single helix is made up of polynucleotide chain. Clear? So it is made up of what? Polynucleotide chain. How this polynucleotide chain is formed? A structural or molecular structure when we see. So I am going to explain about that things first of all, then we discuss this double stranded structure. I already told you there are some nitrogenous base. Suppose this is nitrogenous base adenine, this is nitrogenous base guanine, this is thymine, this is cytosine. Because it is not given in the detail in the syllabus, so we discuss only the outline. So when these nitrogenous bases are combined with pentose sugar, just I draw a symbolic diagram for the pentose sugar. So this is what? Pentose sugar. Clear? So when these nitrogenous base combined with the pentose sugar, deoxyribose sugar, they form nucleoside. I already discussed this thing. And when it combined with the phosphate, they form a complete nucleotide. So by this way, the nucleotide of the adenine, nucleotide of the guanine, nucleotide of the thymine, nucleotide of the cytosine. Clear? So these are what? Nucleotide of the adenine, guanine, cytosine, thymine. When these nucleotides are combined with each other, they are combined by the phosphate molecules and they form a bond that is called phosphodiester bond. So when they combine with each other, they form what? A helix or a strand of the DNA. Clear? Whom I represented like this. And the another helix of the DNA is complementary of this. Means this one is complementary. And that is also made up of the same component. Means their component is what? Their component nitrogenous base, pentose sugar, phosphate. Clear? Pentose sugar, nitrogenous base, phosphate. Clear? Pentose sugar, nitrogenous base, phosphate. Clear? Pentose sugar, phosphate, nitrogenous base. You think, and they are connected with whom? They are connected with the phosphodiester bond. So this is one helix and this is another helix. Here it 3 to 5 direction and another one is 5 to 3 direction. Here I put empty these boxes because there is a definite combination. If there in one helix the adenine is present, so in another helix which is complementary strand, in that there is presence of thymine. So it means adenine is complementary of thymine and guanine is complementary of cytosine. Thymine is complementary of adenine. Cytosine is complementary of guanine. And we can represent like this: adenine, guanine, cytosine, thymine, like this, cytosine, guanine, adenine, like this. And Another side, thymine, cytosine, guanine, adenine, guanine, cytosine, thymine. The another thing is the both helix are connected with each other, joined with each other by the weak hydrogen bond. And in between the adenine and thymine there is two weak hydrogen bond. In between the cytosine and guanine there is three weak hydrogen bond. So we can say the adenine is complementary of thymine, guanine is complementary of cytosine. By this way we can say purine is complementary of pyrimidine base. And in between the adenine and thymine there is two weak hydrogen bond, and in between the guanine and cytosine there is three weak hydrogen bond. So by this way, this is the a symbolic structure of the DNA, and this can be considered as what? A molecular structure of DNA. Clear? Now, this DNA is further go for the coiling, and that coiling is responsible for the formation of the nucleosome. How this nucleosome is formed? There is octant, and this is the histone protein. Histone protein is present in the core, so that can be considered as core histone protein, and it is in the paired form, so 4 histone protein present in the center and all are in the paired form. Around this the DNA strands wrapped. So histone protein along with DNA strand is called nucleosome. So what is nucleosome? The DNA wrapped around the histone octamer is called nucleosome. Now this nucleosome further go for the second level coiling. Then they form the solenoid, super solenoid, and finally it's responsible for the formation of the chromosome. Clear? So there is what? When we see the structure of chromosomes, I already discussed, but once again I discuss here. Chromosome. So chromosomes is made up of 2 chromatids. These chromatids are considered as sister chromatids. Okay, so that is what? Sister chromatids. And both chromatids are connected with each other by a point, and that point is known as centromere. This centromere is also called kinetochore. Clear? So we can say the single chromosomes have 2 sister chromatids, and both sister chromatids are connected with each other by the centromere. Clear? Till here any doubt? To the next point. Next point is gene. What is gene? Gene are a specific sequence of the nucleotides of the chromosome which express itself in the form of character or may express in the form of protein, etc. Clear? So gene a specific sequence. Once again a little bit one thing. So I just summarize DNA. DNA is made up of nucleotide. Nucleotide made up of nitrogenous base, pentose sugar and phosphate. And in DNA, the nitrogenous base are inside, phosphate and pentose sugar outside. The bases, they are complementary. Purine base is complementary of pyrimidine, adenine complementary of thymine, guanine complementary of cytosine. And in between them there is what? There is a hydrogen bonds. So these hydrogen bonds, it's a rungs of the, it's a form the rungs of the ladder like structure of the DNA. Now come to the next thing. that is about the meiosis division. Clear? Meiosis division is not given in the detail, but some topic related with the meiosis division is given in the scope of syllabus. The first, I already discussed you in the previous class that meiosis division is a type of division in which a diploid cell divide by the meiosis 1 and form two haploid nuclei or two haploid cell, and further it divide by the meiosis 2 and ultimately one meiosis division produce 4 haploid daughter cells. This is the significance, this is the meiosis division. And where it happen? It's occur in the meiocyte cells. Meiocyte cells are reproductive cells for the formation of the gametes. Meiosis is responsible for the variation. It's also responsible for the evolution. Even the offsprings which is produced of the same parent, they are different, and it is only and only due to the meiosis division. How they become different, how the variation occur, that is only and only due to a process that is called crossing over. This crossing over occur during the meiosis 1. In the meiosis 1 of the prophase 1, so the prophase 1 further completed by the 5 steps, that is leptotene. It is not in the scope of syllabus and not in the syllabus also, but only the name I discuss here. Leptotene, zygotene, pachytene, diplotene, and finally diakinesis. In the pachytene phase of the prophase 1 of the meiosis, there is crossing over. How this crossing over occurs and where it occurs and what happened. The crossing over occurs between homologous chromosome. Homologous chromosome means the chromosomes, the pair of chromosomes which is exactly similar in gene number, gene sequence, morphology, and one from mother and another from father. So the two, a pair of chromosomes in which one is parental and another one is maternal, fraternal and maternal. So this pair of chromosome is considered as homologous chromosome. And the chromatids of one parent, chromatids of one chromosome and chromatids of another chromosomes. So these two of the same, they are considered as what? Sister chromatids. But when we talk about these two chromatids, one of the one chromosome and another of the one chromosomes, so they are considered as non-sister chromatids. Clear? So first thing, the crossing over takes place between the non-sister chromatids of homologous chromosomes. Where it occurs? The crossing over occurs between the non-sister chromatids. Non-sister, one chromatids of this chromosome and another chromatids of this chromosome. So non-sister chromatids of what? Homologous chromosome. Homologous chromosomes, the chromosomes which is similar in shape, size, structure, gene, and one from the fraternal, one is fraternal and another is maternal in the pair of chromosome. So how it occurs? During the crossing over, the two homologous chromosomes comes closer. Clear? Just a symbolic diagram I draw here. This is one, and they just cross over at a point to each other. The non-sister chromatids cross over at a point. These are what? Non-sister chromatids. They cross over at a point. This point where they cross over, that is called chiasmata or chiasma. So at the point of chiasmata or chiasma, they exchange their genetical material in equal amount, in equal quantity. Same gene number is transferred between two non-sister chromatids of homologous chromosome. Clear? And then after the crossing over, they get separated and they appear like this. You can see the equal amount of the equal segment of the chromatids are transferred. So this is crossed over part. So from one chromosome, the chromatids transfer to it, and from here to transfer to it. So this is crossed over part of the homologous chromosomes between the non-sister chromatids. So this is what? A phenomenon that is called crossing over. Where it occur? Crossing over occur between the non-sister chromatids of the homologous chromosomes, in which a segment of the chromatids gene is transferred in between them. Clear? And this crossing over is responsible for the variation in the offsprings. It also responsible for the evolutionary factors. It also acts as a evolutionary factor. Clear? Any doubt? This way, so by this way, all the topics which is given in the scope of syllabus of the class 10th ICSE is completed. I hope all of you are able to do all the questions which is given in the exercise of your book. So it is my humble request that try to solve all the diagrammatic questions and terms. If there is any doubt or problem, then we will discuss it later. Okay? Thank you.